Hello everyone, my name is Craig Carter, and today I'm going to be talking about the role of glycoproteins in cell recognition. Overview. Glycoproteins function in the structure, reproduction, immune system, hormones, and protection of cells and organisms. They are more hydrophilic than simple proteins. This allows them to be attracted to water more than ordinary proteins. This also leads to characteristic folding of the protein's tertiary structure. Glycoproteins in plant cells are what allows plants to stand upright. Glycoproteins are also helpful with organ systems communicating with each other. The structure of glycoproteins. A glycoprotein has a carbohydrate attached to it. This carbohydrate is used in an oligosaccharide chain, which is covalently bonded to a polypeptide chain. A glycoprotein can be categorized according to the attachment site of the carbohydrate to an amino acid in the protein. There are two types. There is the O-link glycoprotein and the N-link glycoproteins. O-link glycoproteins are ones in which the carbohydrate bonds to the oxygen atom of the hydroxyl group of threonine or serine. N-link glycoproteins have a carbohydrate bonded to the nitrogen of the amino group of asparagine. And to your right is a picture of both the O-link glycoproteins and the N-link glycoproteins. The location of glycoproteins. Glycoproteins are found on the surface of the lipid bilayer of cell membranes. Their hydrophilic nature allows them to function in the aqueous environment, where they act in cell-cell recognition and binding of other molecules. Cell surface glycoproteins are also important for cross-linking cells and proteins to add strength and stability to a tissue. And to your right is an image of the cell membrane, and glycoprotein is highlighted with the arrow above it. These are diseases that involve glycoprotein. So blood clotting depends on glycoproteins prothrombin, thrombin, and fibrinogen. Glycoprotein 6 deficiency is a bleeding disorder associated with a decreased ability to form blood clots. People with glycoprotein 6 deficiency cannot form blood clots normally, which causes them to have an increased risk of nosebleeds. This may cause them to experience abnormally heavy or prolonged bleeding following minor injury or surgery. Women with glycoprotein 6 deficiency often have heavy or prolonged menstrual periods. In some affected individuals, spontaneous bleeding under the skin causes areas of discoloration. Recent findings regarding glycoproteins. Unnatural monosaccharides such as azido sugars that can be metabolically incorporated into cellular glycans are currently used as a major tool for glycan imaging and glycoproteomic profiling. As a common practice to enhance membrane permeability and cellular uptake, the unnatural sugars are per-O-acetylated. The authors developed 1,3-diesterophite in azido-acetyl-galactosamine as next-generation chemical reporters for metabolic glycan labeling. Both 1,3-diol-acetylate and 1,3-diol-propanolated exhibit high efficiency for labeled protein O glycosylation with no artificial glycosylation. Applying 1,3 I don't know how to pronounce it. In mouse embryonic stem cells, they identified what the authors were able to identify ESRRB, which is a critical transcription factor for pluripotency, as a O-glycinated protein. They were able to show that ESRRB O-glycination is important for MESC self-renewal and pluripotency, but catalytically, ESRRB is O-glycinated by O-guanidinic transferase at serine 25. It stabilizes ESRRB, promoting its transcription activity and facilitating its interaction with two master pluripotent regulators, OCT4 and NANOG. So my question regarding glycoproteins is, since glycoproteins are important for reproduction, is it possible to immobilize them in sperm cells to prevent pregnancy? And thank you for listening to my presentation. Hope you guys have a wonderful day. Thank you.